Hello students, in our last class we were discussing about a very important topic, that is enzymes. Yes, so what mean by enzymes? Yes or no? And the most important sentence is that all enzymes are proteins, but not all proteins are enzymes. And there is one sentence, all enzymes are proteins, but not proteins are all proteins are enzymes. Here some of the enzymes are there, they can perform the function of enzyme, but they are not made up of proteins, called as ribozymes. They are nothing but they are DNAs and RNAs. So this is one. And whatever the protein is going to form, that is in the form of primary structure of protein, so that undergoes some structural modification and become secondary and tertiary structure of proteins, yes or not, to perform the biological functions. And one thing is, during structural modification, the protein itself, it will coil itself will become coil. So during that time it will make some pocket like structures called as active site, where the enzyme binds, So, and we were discussing in the, we are in the part that is nature of chemical reactions. See, in that first one was physical change. First one was physical change, nothing but the substrate is being converted into another form, or only the structure is going to change without breaking the bonds. This was first one, what did we discuss at this point. And second one, that is change in state of matter. Already we discussed about this one, that is change in state of matter, nothing but when we have a water, that is in the liquid form. When water become ice, it turns to solid state. Again, when the ice become vapor, it will become gas. So nothing but change in state of matter. This is one. Next, third one is during chemical reaction. Sometime what happens? Forming and breaking the bonds, most important. Yes, during chemical reactions, sometime the bond is going to form and sometime the bond is going to break. So nothing but together, nothing but again there is a what? The chemical reactions. See what kind of the formation and breaking of bonds is going to take place. See, we have AB. Imagine a substrate AB plus C, which gives, what is going to happen? AB plus C, which gives is BC plus A. Yes or no? BC plus A. See, the thing is AB. Yes or no? So here B, the break is going to break. Yes or no? So the bond is going to break between A and B. So here the substrate A, there is a B substrate, in between there is a bond. During this reaction, the bond is going to break between A and B, and in the products, the bond is going to form between B and C. Yes or no? B and C, and A is going to get separate. This is what? The formation of bond and breaking of the bonds. Here it is a breaking of bonds, and here it is forming of bonds. Very simple to understand. And with one separate example. Is BaOH twice plus H2SO4 sulfuric acid, which gives, very simple to understand. Here BaOH twice plus H2SO4 become BaSO4, that is barium sulfate, BaSO4, and remaining is 2H2O. Yes or no? Here breaking of bonds, and here there is a forming of bonds. This is again one of the, the chemical reaction taking place here. I hope you. Next, third one, fourth one, rate of reaction. Fourth one is rate of reaction. So what it mean? When we have, simple thing to understand, when we have a substrate, means any material which is converting into product is called as substrate. So this substrate is converting into product. Yes, substrate is converted into product. At what rate? At what rate? I mean to say, how much time it is going to take and how much products are going to form. So that relation can be written as, that is, rate R is equal to delta P by delta T. Nothing but rate of reaction. Rate of reaction is equal to, is the amount of product formed. Very simple one. P refers to product. So the rate of reaction is equal to amount of product formed divided by time taken. How much time it is going to take to convert the substrate into product. The rate is nothing but R. See, this is what the relation. Next, so likewise we have many examples. We have many examples to justify this statement. How? See, first example I can tell you people. Simple example. In our body, what? We have carbon dioxide. Nothing but when we take oxygen, that oxygen is being converted into, or it is acts as a raw material and being converted into carbon dioxide. So that carbon dioxide mix up with the water molecule. It will become, yes, definitely, H2CO3. Yes or no? H2CO3, right? Yes. In that, this is what? Carbon dioxide, and this is water molecule. Together, you will be having carbonic acid. It will become carbonic acid in the body, as we know that. See, the same reaction is going to take place in our body every time. But thing to understand is, here the thing is rate of reaction. Rate of reaction during in presence of enzyme. Rate of reaction presence of enzyme. See, what is the capacity of enzyme? Here we are talking about the capacity of enzyme. I mean to say that the same to same reaction, that is CO2 plus H2O, which gives carbonic acid. The reaction or this reaction is going to take place, that is 200 molecules. 200 molecules of H2CO3. Yes, 200 molecules of H2CO3 are going to form per hour. Getting my point? Per hour. See, in 1 hour almost there are 200 molecules of carbonic acids are going to form. Yes, but thing is that same reaction is going to take place in presence of an enzyme. Yes, same reaction is going to take place in presence of an enzyme called as carbonic anhydrase. Enzyme yaudri? Carbonic carbonic anhydrase. So in presence of carbonic anhydrase enzyme, the same reaction is going to take place, that is 6 lakh molecule. 6 molecules of H2CO3 per every second. Can you imagine this? Yes, that is the power or the capacity of enzyme, that is carbonic anhydrase. I told you in the last class, that is usually always enzymes ends with the ASE. So carbonic anhydrase enzyme. In presence of this enzyme, there are 6 lakh molecules of H2CO3, that too for every per second or every second. But in absence of this one, only 200 molecules per hour. So this is what the rate of reaction in presence of the enzyme. I hope you. Yes. And one more type of the chemical reaction. One more type of chemical reaction is multi-step reaction. One more is multi-step reaction. See, in multi-step reaction, what mean by multi-step reaction? I mean, see, as I told you many times, when we have a substrate in presence of an enzyme, substrate is being converted into product. Yes or no? This is what? A single step reaction, whatever you take. See, what we are discussing right now is, sometimes we have a substrate, nothing but I can write like S1, means first substrate or substrate first. See, in presence of an enzyme, it will become S, that is S2, that is second substrate. Is already we discussed na, that is metabolic pathway. It is like what? Turnover. I mean, A is converted into B is called C into D into Z, called as metabolic pathway. So here also S1, first substrate in presence of enzyme, converted into substrate 2, and substrate 2 is converted in presence of an enzyme, S3, S4, finally it will become product. Is considered as multi-step reaction. So for example, we have, that is, glucose is being converted into 2 molecules of pyruvic acid. Clear? So the example is, glucose is being converted into 2 molecules of pyruvic acid in presence of enzyme, no doubt, but it takes place in 10 steps. That is, say for example, glucose converted into glucose 6-phosphate, glucose 6-phosphate into fructose 6-phosphate, fructose 6-phosphate into fructose 1,6-bisphosphate. Likewise, there are 10 steps right from glucose to 2 molecules of pyruvic acid is nothing but multi-step reaction. Hope you understand. So in a series of the steps are involving, but all steps are going to involve the enzyme. It is mandatory. Enzyme is mandatory to convert the substrate into product. I hope you understand the whole concept. So what to understand? Headline is chemical reactions. Is in chemical reaction, what kind of reactions are there? First one, physical change. Without breaking the bonds, only the structure is going to change. Example, I told you people, square to irregular or round or rectangular or whatever you take. Second, change in state of matter. That is, example, water, ice, vapor. Forming and breaking of bonds, that is BaOH2 H2SO4, BaSO4 plus 2H2. Then one more is rate of reaction in presence of an enzyme. So R, how to read this? Rate of reaction, or we can call it as, better word is velocity of reaction. So the velocity of reaction is amount of product formed and divided by time taken. This is what? Example, CO2 plus H2, it is H2CO3 called as carbonic acid in presence of carbonic anhydrase, in absence of anhydrase. So this is what? 200 molecules, 6 lakh molecules. And last one is multi-step reaction. Is next we have one more topic. How does the enzymes can bring the rate of reaction? How does the enzymes bring what? How does the enzymes bring the chemical reactions? How does? See, what is the strategy here? What does the enzymes can bring this much of chemical reactions? See, before this one, we need to understand. First one is when substrate binds to enzyme, or else we can write like when substrate is being converted into product, first and foremost. Yes or no? Substrate is converted into product. In that, second one is substrate in presence of enzyme which gives product. This is what, again, one more standard. Substrate first binds to enzyme, later the enzyme can break or it can form the bond for the formation of product. See, this is what. In that, one more is, first word is substrate. What mean by substrate? I told you many times. What is substrate? Any material which is converting into product is called as substrate. It may be sucrose, it may be lactose, it may be what? Complex protein molecule, carbohydrate molecule, whatever you take, all come under substrate. Second one, active site. Yes, I told you again many times. What is active site? Where the substrate binds to enzyme. Where substrate binds to enzyme. At what location or at what position substrate hold the enzyme is called as active site. Clear? It is present on the enzyme always. Next, what happens? First I can tell you the whole sentence. How? See here. First we have substrate. This substrate initially binds with the respective enzyme, not with all enzyme. I mean, we have a sucrose. Sucrose always binds with the sucrase. Maltose, maltase. Protein, protease. Likewise, substrate binds with the enzyme, respective enzyme. First, it can make enzyme substrate complex. Remember the word. When substrate binds to enzyme, first it can make enzyme substrate complex. After the formation of enzyme substrate complex, enzyme starts converting the substrate into product by either breaking the bond or by forming the bond. So it will become enzyme product complex. Enzyme product complex. After the formation of enzyme product formation, I mean, see, the thing is, product has been formed, yes or no? But thing is, still there must be a close relation with the enzyme. So that finally enzyme plus product. Over. Can you remember the whole sentence once again? How to read? See, when we have a substrate, it can bind with respective enzyme. It leads to formation of enzyme substrate complex. Clear? Yes. When enzyme substrate complex leads to enzyme product complex, finally the enzyme can be released from the substrate. I can give the example. See, when we have an example, there is substrate S. Is when we have substrate, same time when we have enzymes, nothing but, yes, this is enzyme. Now what happens? Enzyme substrate complex means yes or no? This is called as enzyme substrate complex, means substrate binds to active site of enzyme. Now enzyme product is formed, nothing but this is what? Product. There are two products. And finally, enzyme plus over. This is what? When you take example, we have sucrose as a substrate. It binds to sucrase. It will become sucrose plus sucrase complex. Yes or no? Sucrase complex. Later it will become sucrase plus. Yes, what is going to take place? Glucose plus fructose. Because glucose and fructose together, sucrose, it is going to break the bond between glucose and fructose. Later they become sucrase. Finally, sucrase plus glucose plus fructose. These are nothing but products. Clear? This is what? The simple thing to understand. Next, when we have how does this reaction takes place exactly? Yes or no? So we'll discuss here. See, in one side, during enzymatic reactions, the word is during enzymatic reactions. What is going to take place? See, during enzymatic reaction, when substrate binds to enzyme, see, it particularly binds to enzyme. So now the enzyme is about to break the substrate. When the enzyme is about to break the substrate, so that time the substrate is going to change its state, or the structure is going to change. Yes or no? The structure is going to change. So where, at what point, particularly at what point the structure is going to change is considered as transition state structure. Please do remember the sentence. That is, when enzyme substrate complex is going to take place, during that time the enzyme starts breaking the substrate. So at what point the substrate is going to break? Yes or no? See, one thing you have to remember, at what point I have written here, that is transition state structure. So at that point still the substrate is remain as a substrate, it is not converted into product. So for that reason consider as transition state structure. See, I can give you by one simple experiment. Here we have one stick. We can take an example of matchstick. See, here we have, it is considered as a substrate, and after conversion of product it will become 2-piece. The 2-piece nothing but product. Yes or no? See, when we apply some pressure with hand, so now the pressure with hand nothing but enzyme. See, when I hold this stick to break, yes or no? Because when I break this, it will become 2-piece. These 2-piece nothing but product. To form product, I need to break this stick, so that when I hold this stick at this position, yes or no? So when I start putting the pressure, when I start bending this, so at what point it will break at the middle to become 2-piece? Now at that point the straight stick become like this. Am I right or wrong? So now during, at what point the substrate is being converted into product, so during that time it can change its shape, is called as transition state structure. So very immediately the transition state structure is being converted into product. I hope you understand. This is what, how does the enzyme can bring high rate of reaction. See, this is one thing. Hope you understand my point. What? Here we have a substrate. To convert it into product means we need to put some pressure. So, or we need to use the enzyme. Yes. So at what point the enzyme can convert a substrate into product means, so there it is going to change its structure. So at what point it is going to change its structure is called as transition state structure. So very immediately the transition state structure is being converted into product. Hope you understand. And last word is that activation energy. Last word is activation energy. So now tell me, what is activation energy? By understanding this concept, we can tell that activation energy means the energy difference between the transition state structure to product. I mean, to better word is, see, when we have what? Imagine it is a what? Substrate. When I use my active site, yes or no, enzyme active site, now it is going to convert into product, means it become 2-piece, yes or no, 2-piece. So to become 2-piece or to make 2-piece, I need to put some pressure, yes, like this. So now at what point the structure is going to change? I mean, when it is about to become 2-piece. It is about to become 2-piece means it is not converting into 2-piece. It is about to convert it into 2-piece. So at what point it can change the structure is called as transition state structure. Yes or no? So to make it 2 piece, how much energy I should put is considered as activation energy. Clear my point? It can be shown by some graphical representations like this. This is what? The enzyme. I mean activation energy. I mean transition state structure. So this is what? The potential energy. And here what happens when I start putting the pressure at certain point, nothing but certain point refers to the substrate is about to convert it into product. So that time it will become 2 piece, nothing but product is formed. And whole sentence can be is substrate binds with enzyme, enzyme substrate complex is formed. Then enzyme start breaking the bonds between substrate, or it can forms bonds. Now it will become enzyme product complex is formed. Finally, after formation of product, enzyme can be released from the product. Finally, product is formed. This is what, how does the enzymes can bring high rate of reaction. Yes, and remaining things we'll discuss in tomorrow's class. Thank you very much.